tissue talks. As a reminder, please use the Q&A feature for any questions you have, and we'll answer these questions at the end of the talk. And we're really excited today to have Dr. Thomas Eschenhagen here from the University Medical Center Hamburg-Eppendorf to join us for our second to last tissue talk of the spring season. And so Dr. Eschenhagen is a professor of pharmacology and director of the Department of Experimental Pharmacology and Toxicology at the University Medical Center in Hamburg. And since 2011, he's also been the speaker of the German Center of Cardiovascular Research. Dr. Eschenhagen received his MD in pharmacology and toxicology in 1988, and then after residency, he received a prestigious Heisenberg stipend of the German Research Foundation and spent numerous short-term fellowships at the Washington University School of Medicine, Stanford University, the National Institute on Aging at the NIH, and the INSERM unit in Paris. In 1998, he became chair of the Institute of Clinical Pharmacology at the University of Erlangen in Germany, and then in 2002, he took his current position in Hamburg. He is a member of the German Academy of Science Leopoldina and past president of the International Society for Heart Research, finishing his term in 2022. In addition, he's been elected a fellow of the American Heart Association, the European Society of Cardiology, and the International Society of Heart Research. Dr. Eschenhagen pioneered the engineered heart tissue that many of us in the bioengineering field are very familiar with in 1994, which in combination with human embryonic and now induced pluripotent stem cell technologies can use pluripotent stem cell derived cardiomyocytes to develop medium throughput tissues for both drug testing and for disease modeling. The development of these tissues for disease modeling also helps support Dr. Eschenhagen's group in a European Research Council Advanced Grant that he's had since 2014, which has supported his group's effort in studying these mechanisms of both heart failure and then also the application of engineered heart tissues for regenerative medicine. Dr. Eschenhagen's group has published over 400 papers with over 40,000 citations and an H-index of 106. And so we're really excited to have him here to speak about his work on human iPSC-derived cardiomyocytes and engineered heart tissues for modeling cardiac diseases. Where do we stand? So thank you so much for joining us, Professor Eschenhagen. You can now take over the screen.
Great. Thank you very much. This was a very kind intro. So I'm happy to be here and speak about iPSC and engineered heart tissue. And it's actually the second time I speak about speak here in this seminar. And remember, the first time it was really very it was nice. So I hope it's going to be nice today. What I wanted to do today is to share a little overview, of course, but then show you some still unpublished data on which we have actually worked on for many years. Do you see my screen well?
Yep.
Great. So I'm going to use the laser pointer. So before starting, just a little disclosure. I'm a scientific advisor and shareholder of Dynacor, which is a gene therapy company. I'm on the board of directors of DynaBios, which commercializes the EHT technology, and I have a strategic partnership with Evotec to develop a cardiac repair approach. So I guess all of you know about the promises of the iPSC technology. It is giving us unlimited supply of human cells, and in our case, it's cardiac myocytes mainly, but not only. It provides a test bed for drugs and gene manipulations in a human context, and it's an excellent system to model human cardiac diseases. And of course, as an important side, it also reduces animal experimentation. And when Yamanaka got his Nobel Prize 2012, one of the promises was also to use these cells to regenerate diseased organs, and we're working on that, but I'm not going to talk about this. So if you look in PubMed for iPSC and cardiac diseases, the field had a steep increase until early 2020s, and now it's somewhere around 300 to 350. So I think it's fair to say the iPST technology is already an integral part of cardiovascular research. And I believe it will increasingly be also an integral part of pharmaceutical drug development. And one of the important milestones here was this FDA ruling in the end of 2022, which essentially said we do not necessarily need animal experiments for all regulatory issues. It doesn't actually say we don't need animal experiments anymore, and some people somehow mistranslate this, but it says there is room for new systems. And there was a recent paper, which is actually very readable, a roadmap to reducing animal testing, and their organ on a chip systems, computational modeling, and advanced in vitro assays play a pivotal role. So far to the promises. I think you all are also aware of certain limitations. Cardiac myocytes from iPSC are still immature. There is a somewhat limited functional readout if you compare this with a whole animal. And the robustness of the in vitro phenotype is an issue. And finally, variability and several problems with these cells do present a limitation I'm going to talk about a little. So if it comes to immaturity and functional readout, as Navid said, we have been developing these engineered heart tissues over the last actually 3 decades. And you see here the timeline. I'm not going to go in all the details, but it started actually really in '94 with this biconcave lattices, at that time from chick cardiac myocytes, and now we're using this automated system, and I think many others do this as well. So a bit closer look about on the development of these engineered heart tissues. You see, this was the original format. These were lattices spanning between 2 Velcro-covered glass tubes, which were held on distance by a metal brace. This worked actually quite well, but it was not good for higher throughput. We then developed this ring format system, and now the system which is easily multiplexable, where the EHTs is spanning between 2 flexible silicon rods and forms a tissue strip, which is then, and I think that is an important issue here, contracts against an elastic force. So it always performs auxotonic contractions, very similar to what a heart does. And in the meantime, many other groups developed systems. I know your group in New York did as well. And this looks similar, but there are certain modifications. Others have produced higher throughput systems. So overall, the technology really became used worldwide. And of course, it's kind of rewarding to see this. So why is it an advantage compared to immature or 2D cells? If you look at the histology, and this is just a section through an EHTs, in green are the cardiac myocytes, in red are connexin 43. You see, this is a real heart tissue. Look at the nuclei, they're elongated, which is a typical form of more mature cardiomyocytes. It's a dense tissue, well-oriented. And I remember the early days of iPSCs, where people started to measure action potentials in 2D cardiomyocytes. And they always reported a very low membrane potential, meaning a depolarized membrane potential, about -40, -50, and a very low upstroke velocity. If you do sharp microelectrode recordings, as you see here on the left in red, of 2 EHTs from different clones, you see the membrane potential is around -80, this is physiological, and the upstroke velocity is 250 volts per second, which is actually exactly the same as here the native tissue, which you see on the right side. So this looks actually quite good. We then, some years ago, compared the proteome profile from 2D cells and EHTs. And here's a comparison. the Volcano plot. You see here nuclear proteins, in the middle mitochondrial proteins, and the right ribosomal proteins. And you see clearly the most evident change or difference between these formates was that there were more mitochondrial proteins in the EHT. So we thought, okay, maybe these cells really also have a different metabolism. So we measured this directly and found that if you compare the substrate preference of 2D cardiomyocytes on the left and 3D in the middle, you see that in orange here, the fatty acid oxidation is only 40% in 2D, but it was 65% in 3D, and the glycolysis, in contrast, went down from about 50% to 14%. And that is not exactly as adult cardiomyocytes, but it's coming close. So again, a clear indication that in the 3D format, cells become more mature. So I think immaturity is not solved completely, but I think it's a big step forward. And obviously, in these EHTs, you have a very nice functional readout. What about robustness? That is an issue. It's easy to multiplex EHT. So this is a view in one of our incubators. So we always have hundreds of EHTs here in these 24 well plates in culture. So it's relatively easy to make numerous EHTs. And some years ago, we simply observed these EHTs over about 20 hours or so. What you see here, these are measurements every half an hour overnight. You see force development in blue, frequency in green, time to peak, and time to relaxation. You see these parameters are really stable over many, many days, even weeks, sometimes even months. So the system is robust and multiplexible. But what about variability? And I think there are lots of factors of variability. There's obviously variability between donors. There's experimental variability, karyotype problems, pluripotency problems, and clonal shifts, and there are many others. And if you look at the procedure you are all well aware of, starting with a skin biopsy, fibroblast culture, reprogramming, clonal selection, passaging of cells, master cell banks, and then cardiac differentiation protocols to come finally to cardiac myocytes and/or EHTs. That takes at least 80 days, probably realistically even longer. So some years ago, we asked the question, are iPSC myocytes and EHTs really feasible as a screening platform for individual cardiac phenotyping and risk prediction? I came to that question as well because clinicians started to think about making thousands of iPSC lines from these big clinical studies to get a better handle on the phenotype of the patient. So is it really possible? And specifically, we asked the question, can we detect group differences, disease group differences in a blinded manner? So without knowing anything about the cells, just straightforward in a blinded manner. And is there something like a genotype-phenotype correlation? Initially, we also wanted to ask the question, does the in vitro phenotype predict the prognosis? But we came quickly to the conclusion that that is probably too ambitious. And at that time, we chose two diseases, dilated cardiomyopathy and hypertrophic cardiomyopathy, as prototype cardiac diseases, which clinically present with the opposing phenotype and both have a strong genetic component. So this was the background or the goal of this ERC project, which I think Navid also talked about. So the European Union gave me a lot of money for this project to finally have a bigger group of healthy probants. We came up with 39, almost 40. And in the end, we had 8 patients with dilated cardiomyopathy and 12 with HCM. The clinicians did the cardiological examinations. We did panel sequencing, and then they did a skin biopsy and gave us a tube only with a barcode. So we were completely blinded. We made fibroblasts, we made the entire procedure, made EHTs, and then measured basal function, long-term rhythm, the response to several drugs, and RNA sequencing. I'm going to show you some of the data. The overview looks like that. Again, this is a procedure, a bit at more detail. I think I don't need to go through all the details. Maybe just to give you a flavor of the amount of work. So you see we made 83 master cell banks, 2,000 wide working cell banks, 128 cardiomyocyte production batches, and in the end we came up with more than 1,000 EHTs. And that took, in average, 189 days, so quite a long procedure. And then the EHTs were subjected to a SOP-controlled program of functional tests. So we tested at around 15 to 18 days the response to low extracellular calcium, I mean the force response. Then we tested force frequency relation, post-rest potentiation, and a few days later, we challenged them overnight with high calcium. Then next day, we went back to low calcium and then applied isoprenaline. And then in the presence of isoprenaline carbachol, and then yet a few days later, we tested the effect of the calcium sensitizer EMD and the hERG blocker E4031, and finally we shock froze the EHTs. Some data. So this was the genetic analysis. So we subjected them all to panel sequencing. It's 172 cardiac genes which are sequenced. And here are the results. In green, no variant. In yellow, variants of unknown significance. In red, pathogenic mutations or likely pathogenic. And as expected, of course, DCM had about 50% pathogenic mutations, HCM a bit higher. But surprisingly, also the control group had two variants, pathogenic variants. So this also has to be taken into account if one works with so-called healthy controls. We then looked for, in the end, of course, we evaluated, so how successful was all what we did, the quality of all the clones. Here we looked at 80 clones, and you see the composite quality control was only good in about 65%. Some had a smaller problem, and about 1/4 had a major problem. We had quite some karyotypic problems. Red is duplications, trisomies, and this pale color here is some mosaics. Sendai virus clearance was there in about 80%, and overall EHT quality was good in about 70%. So this already shows you that it's not easy to work with such an amount of cell lines, and that you cannot expect to get 100%, even though the group doing this was really very experienced. So these are the mean data here. We used SSEA3 as a pluripotency marker, and you see in average is very good, it's 95% and more, but there were also always some outliers. And the same holds true for TNT positivity, so the percentage of cardiomyocytes, in average about 75 or 80%, but again, quite some outliers. If you look at the overall success rate, now in terms of from how many probants did we finally get good EHTs? And if you look here on the left, these are the controls, in the middle is the DCM, and interestingly, the HCM success rate was more than 87%, while DCM and control were only about 56%. So this was actually statistically significant. For some reason, the HCM worked better than the rest. Was it due to the mutations? That's the obvious question. And here I show you data, either black, there's nothing, so no variant. Yellow or orange is variants of unknown significance, and red are the pathogenic mutations independent of the group. And you see there was a significantly higher karyotypic problems in those with the mutations, but the rest was independent of the mutation status. So this is sender virus clearance, TNT positivity, and EHT quality. If you ask how is it according to the disease to the disease group, now it's a control DCM HCM. There were no major difference with one exception. The TNT positivity was lower in the DCM lines than in the rest. Okay, so this in a way maybe was expected, but the better performance of the HCM lines was clearly not expected. So now we looked at the EHT function, and I'm not sure how much most of you are familiar with the EHT system. So what you see here, I go through a little bit slower. What you see here always are contraction peaks over time. These are in general 20 seconds. And what you see here in the upper, this on the left is a normal behavior. That's what we are used to. This is the ERC12 line, for example. This is spontaneous beating, very regular, at 1.8 millimolar calcium, no pacing, looks like that. If you pace, you see that it nicely takes the pacing. Then if you go down with the calcium from 1.8 to 0.6, you see the peak gets smaller. If you add isoprenaline, the force increases. And then if you look here, this is the effect of the calcium sensitizer EMD, and this is E4031. You don't see this here probably in this, but if you look at the single peaks, you see here the difference between spontaneous beating and paced beating. So the spontaneous is wider, the paced is smaller, and then depending on the calcium, it just goes down. Isoprenaline makes the peak smaller, I mean shorter. So this is the classical positive inotropic. This is the increase in force, but also positive lusitropic effect. It's a bit shorter here. You don't see this well. In contrast, the calcium sensitizer EMD makes contraction a little bit bigger, but much wider. And that is actually an important point also for clinical use of these drugs. They impede relaxation, so they make relaxation longer. And what you see here is also typical with the E4031. Blockade of repolarizing IKR currents lead to a prolongation of terminal relaxation, and that leads to this buckle here in the end of the contraction in the relaxation peak. So this is a normal behavior, which we see in the majority, but then you see just examples how it could also look in this system. So this is spontaneous arrhythmia. You see here this very wide peaks under low calcium, or here a dramatic increase in contraction time under EMD, or here after depolarizations under E4031. So these are typical abnormalities seen in different lines under different conditions. And of course, the question was, are these pathologies somehow restricted to certain genotypes or certain diseases? And the answer is not really. So this is now EHT quality and function. So you see here always the three groups: green control, red DCM, and yellow HCM. I try to keep the color code overall. And you see here again, category 2 means they have some problems. And again, DCM looks worse than control, and HCM looked actually perfect. There was nothing. The HCM lines were from this parameter perfect. Arrhythmias were more seen in DCM and then both other groups. EAD, so this early after depolarizations, were again seen more in DCM than in control, never in HCM. Fraction of lines with EHTs showing biphasic relaxation were for whatever reasons, lower in DCM. And here you see the only thing which looked pathological in the HCM, namely the fraction of lines with EHTs showing white peaks when unpaced. So this seems to be, could be an HCM phenotype in this system. We quantified, of course, force parameters. Where is this? I don't know where this. Oh, here. Here's spontaneous rate, RR scatter, so the parameter of arrhythmias, and force. And here you see the expected picture. Force was lower in mean. The mean force was lower in DCM than in control, and no difference HCM. RR scatter, arrhythmias, you saw this already, is higher. In DCM was higher, and the rest, here's a small, longer time to peak tension, but the rest was the same. And you see, of course, that the overlap of the group was really big. So here, for example, force, you see even in the control group, there were some lines which were really very low force and others were perfect. So this shows already that an individual risk prediction, as we have hoped, is at least with this approach not possible. We went on and made some more drug testing. Somehow my computer. Yeah, now it's came. So this is relative force under isoprenaline. I guide you through. If you just look at the controls here in green, at 0.6 millimolar calcium, they developed about 70% of the force of 1.8 millimolar, which is normal, but they go down. If you then apply isoprenaline, there's about a 30% increase in force. If you compare this column here of 1 millimolar calcium baseline and then 1 millimolar plus EMD, there's about a 20% increase in force and a decrease in force with E4031. And in principle, this looks all the same in all three groups, except that DCM is always on a lower level. But you see the rest, there were no qualitative or quantitative differences. They in principle behaved all the same, just that the baseline force and actually the reaction to calcium was stronger in the DCM line. And the times didn't give much of a answer here. They looked actually all the same, the time to peak and relaxation time more or less the same, which we found unexpected because we had expected that, for example, the HCM have a prolonged relaxation time, but this was not seen in the statistics. So then we tested the force frequency relation. You see actually just as an example, we normally made 8 EHTs per batch. And you see they look pretty much the same. Also the beating of these EHT is pretty much the same. So the EHT system itself is not much of a source of variability. It's everything before, in my view. So you see they look one like the other. And then the force frequency relation is a typical one. You see they take the pacing in this case at up to 3 Hertz. Then sometimes there's a lack of pacing, or here up to 4 Hertz, and then force goes down. But between 1 and 1.5, there's a slight increase in force, and this is also shown in the statistics. This is a normal behavior here. This is a capture rate. This is the force. You see there's a biphasic relationship. First it goes up and then it goes down. And it's always remarkable to see the time to peak tension, time to peak and time to relaxation. There's such a strong effect on the contraction kinetics. You see this is not even a third, or it's about a third of the time at 0.75 Hertz with time to peak. Was there a difference in force frequency behavior between the different groups? somehow my computer. Ooh, oh. Ah, okay. Do you see the screen again? I hope so. So you see here, control positive frequency relation was more than in DCM, but again, the HCM looked better or had a clearer false frequency relation than control and clearly more than DCM. So the outlier again was DCM. So to summarize this data, what does it mean? I mean, first of all, I think this is the first prospective blinded clinical study with iPSC cardiomyocyte, at least the only one I'm aware of. The overall success rate was 50% in DCM and ranged up to 92% in HCM. There were relatively consistent group differences, DCM worse, HCM rather better than control, but there were no clear genotype-phenotype correlations and overall a large variability. So which makes me conclude that at present, this technology is not suitable as a screening assay for risk prediction. So I don't want to leave you with a slightly disappointing message. Of course, there are many, many examples of successful disease modeling, but I underlined here dedicated disease modeling. So I think it is a bit naive to think that you can simply make, and that was our project showed, make 100 iPSC lines and clearly see in a relatively small number of possible ends effect. So I show you an example where we did clearly find something interesting. So we were interested in this phospholamban cardiomyopathy. It's a very popular mutation, particularly in the Netherlands, this R14del. And until then, most people believed that the defect is about calcium excitation contraction coupling. So we made this iPSC, we made isogenic control. You see here very large N numbers. In blue are the isogenic controls made by CRISPR, and in red the patient line. And overall it was clear the patient line developed less force, which you would have expected for a dilated cardiomyopathy cell line. Here is the average peak. It's pretty clear. We then looked at calcium cycling. So we measured not only force, but also calcium cycling. And surprisingly, the calcium amplitude was exactly the same as in control. The only difference actually which we found was a very slight prolongation of the peak, but amplitude, diastolic calcium, all the same. So this did not argue for SR calcium cycling defects. So we looked on, and for example, we found these pictures here. So in electron microscopy, you see left on the left an isogenic control with nice mitochondria, a normal endoplasmatic reticulum and one sarcomere, and on the right the nucleus. And in the phospholamban mutation, we found blown up ERs a lot with filled with lipids, very pathological endoplasmatic reticulum. So this, and we found many other things. I don't want to go into too much detail here, but this paper provided evidence that in fact the main problem might not be defective excitation contraction coupling, but ER dysfunction. And rewardingly, more or less the same time, Mark Mercola's group in San Diego showed in a circulation paper pretty much the same, and they found increased ER stress. So these papers really changed the field to a different understanding of what's going on in phospholamban R14del cardiomyopathy. A second example is, in my view, interesting as well. This is done in the group of Lucie Carrier in our lab. She worked together with a clinician who found a patient with the HCM. This is the index patient here. And they found in this patient with a very severe form of HCM and a septum myectomy, they found a heterozygous actinin 2 missense mutation. And when we looked in the databases, this was shown as a VUS, a variant of unknown significance. So we did iPSC. Here's a control line, the sequencing. Here's the HCM line. And you see here's the mutation. And then we corrected it by CRISPR-Cas. So we had 3 different lines here and did EHTs as usual. And found something, but I cannot show it because my computer is here. So first of all, Max Bronczynski, who did these experiments, found that these HCM cells were bigger, the cardiomyocytes, which you see here compared to controls. Here's the statistics. Second, he found myofibrillar disarray, which is again something relatively typical for HCM. But then he made EHTs. And what you see here in red, this is the HCM EHTs compared to black control and gray isogenic control. And you see here a marked increase in force where this is maybe not also normal, but then this marked prolongation of relaxation. And we thought, okay, maybe that's due to the well-known increase in calcium sensitivity, and actinin is a sarcomeric protein. So this was something which would have made sense. And in fact, we did see a little shift to the left compared to the control. So there is indeed a slight calcium sensitization of the myofilaments, as it is typical for HCM. However, we said that cannot be all. And so we made action potential recordings. And here we saw something really interesting, namely that the action potential was markedly prolonged compared to both controls. And that actually let us measure calcium currents. And indeed, the calcium current, again, red versus black and gray, was more than doubled in these HCM lines. And then we asked, what is the relation between calcium channels and actinin? But there is actually literature on this, showing that actinin is probably organizing the calcium compartment. In any case, we went on and said, okay, if the increase in calcium current is an important player here, we should be able to normalize the phenotype by treating with a calcium channel blocker. And that's exactly what you see here. So when we put diltiazem, an L-type calcium channel blocker, on this HCM line, we saw a marked shortening of the action potential repolarization, while the effect in control was modest or almost not there. And then we thought, okay, I mean, diltiazem is something which we can give to the patient. So we went back to the patient and we found actually that this patient had a long cure QTC interval, not really very long, but longer than normal. So in fact, it was a QTC prolongation, which the clinicians actually didn't take or didn't see before. So we treated this patient with diltiazem. In fact, we replaced amlodipine by diltiazem. And you see here, QT interval completely normalized. And actually, this was this was stable for several years. So we believe that this is a nice example of a personalized medicine. We don't know really whether this cured the HCM phenotype, but at least it probably helped the patient. And I think it's just an example of what this technology can do. So it's a novel missense mutation. The mutation unexpectedly affects proper LTC inactivation and an AQT syndrome, and the data led to therapy optimization. Another example, a more recent one, we got interested in this famous PITX2 SNP. You probably know that atrial fibrillation is the most common arrhythmia in humans, and this transcription factor PITX2 is, or a SNP in this gene, upstream of the gene actually, is very strongly associated with the prevalence of atrial fibrillation. And nobody really knows what PITX2 has to do with atrial fibrillation. So we thought, okay, let's model this. For this, we needed good atrial EHTs, so we adapted protocols of others and used the retinoic acid protocol and had success. But then we realized that sometimes we had a very nice action potential, which looked like that, but sometimes it looked like that. And in the end, we found that we didn't treat our retinoic acid very well. So you have to be really careful with the retinoic acid concentration, because you see here the concentration dependency is extremely steep. This is an action potential of a ventricular EHT. This is of normal cardiomyocytes. Then if you add in the differentiation protocol 0.01 micromolar, 0.1 or 1 micromolar, you see this dramatic shift in action potential. This gets shorter, and you see here control the APD20 is extremely short. It gets a spike and dome shape, and importantly, it has a very funny and characteristic response to 4-aminopyridine. So point here is you need high concentrations, and you need to be careful that you use really fresh retinoic acid to get the right response. So then we made a knockout of PIDX2 in this iPSC, and you see here the phenotype, the action potential phenotype. In black is the isogenic control, and if you knock out PIDX2, it looks like that. It's a marked prolongation of APD20 and a small shortening of APD90, and here the data to it. The important point is that this phenotype, in fact, looks very much like the respective phenotype of the atrial fibrillation, actually, which I just didn't. Oh no, I'm sorry. This I don't have it, but it really looks like the difference between sinus rhythm and atrial fibrillation in humans. So we are pretty excited about this phenotype, because it means that somehow important ion channels must be under control of PIDX2, causing this pretty dramatic electrophysiological phenotype. And we are presently asking the question, is it really PIDX2 itself? Because we did a pretty big knockout, so we are not quite sure whether these could not also be other genes, and I hope we find the answer. And before ending, I want to show you another example, which I think is important somewhat, because the question is really, how practically important is all this work for drug development? And as I said, I'm involved with Dynacor and the gene therapy development here for myosin binding protein C replacement in patients with hypertrophic cardiomyopathy and truncating mutations in myosin binding protein C. So we did again EHTs, and you see here the phenotype differences between the isogenic control is in dashed black, while the homozygous myosin binding protein C is red. So this is a completely different phenotype from the actinine, as you see. This is not a prolongation of relaxation, but this is a shortening. And it turns out that one important point here is the inability of these EHTs from mutated C proteins to keep systolic ejection. So this is much shortened, and that leads to systolic deficit, not only diastolic deficit. And we use this system as a test bed for AAV-mediated gene therapy for myosin binding protein C3. And you see here this particular C protein AAV completely normalized this contraction kinetics compared to the knockout. And the company BioMarine, which we were collaborating with at that time, used these data, or actually produced numerous variants of these vectors. And you see just examples, BB3, BB6, BB5, and you see they all look a bit different. And the best repair was seen with BB6, while BB5 had no effect, and BB3 had somewhat an intermediate effect. And this is the statistics of this. It depends partially on the protein expression, And on the basis of these data, the company picked BB6 and developed this towards clinical approval by the FDA. So I think it's important because it's one of the early examples that EHTs, in fact, or work in EHTs, in fact, was relevant for an IND approval by the FDA. So with this, I would like to conclude or summarize. I think iPSC have a great potential to replace animal experiments in biomedical research, particularly for drug screening. That's the easy one, preclinical drug safety pharmacology, and also I think for the development of cardiovascular therapeutics. I also believe there can be an important part of personalized therapy concepts, but to do so, one really needs isogenic controls and replication in high numbers of batches and cardiomyocytes and EHTs to really get a valid phenotype. Overall, I would say more standardization and automatization are necessary to reduce the variability. Stress tests are necessary often to unmask phenotypes. And our in vivo hard data indicate that this approach is not yet, at least, suitable as a screening assay for individual risks in patients or families with cardiomyopathy. And with this, I would like to thank many, many people. I cannot list them all. The in vivo heart project was mainly run by Bärbel Ulmer, Aya Shibamiya, Thomas Schulze, Mirja Schulze. This was an enormous effort over several years, so I'm really grateful for them that they didn't give up. We had great collaboration with the clinicians, with geneticists, with sequencers. Dynacor gave some support. There are many other people involved and many funding agents. And with this, I stop and thank you very much for the attention. Hello? Is there anybody? Ah, there's somebody. It's funny, this format. I don't see anybody. Shall I go into the question answer? Okay, let me see. Okay, so I just go through them from the beginning. Let's see how far we come. What quality control was performed on the patient-derived iPSC directly? Exactly, apart from the karyotype. So we did SSEA3 percentage as a pluripotency. We did Zenda virus clearance. We did, of course, STR, which is actually very important to prove identity, and a mycoplasma test. Karyotype, she said already. And the second question, in summary, okay, that's about it. That's what we normally do. So karyotype, Sendai virus clearance, mycoplasma freedom, and STR for identity. The question is actually, maybe you're interested in that question, what is the best method? At that time with the in vivo heart, we did G banding. That's quite expensive and also a bit subjective maybe, even though it was done by our clinicians, so it was, I think, good quality. In the meantime, we do mainly nanostring panels for karyotyping.
Sorry, my computer died, or like it stopped the connection for some reason.
I just go through the comments here. Can you comment on EHT variability even within groups? Could it be due to differentiation protocols variability and maybe, yeah, that's a very good question. We also, I didn't show the data, but we did what we called a 3x3x3 clone project. So we had 3 donors, made 3 clones of each donor, 3 batches of each clone, and I don't know how many EHTs. And the answer is, so to answer the question, where does the variability come from? And the answer is, it comes from the donor, but it also comes very strongly from cardiomyocyte differentiation. And of course, we are looking for differences between donors. So that's actually what should not be excluded. But I think a goal must be to reduce variability of the cardiomyocyte differentiation protocols. Variability due to EHT is actually very low. So if you have a good batch of cardiomyocytes, making EHTs is easy, and they do not vary a lot. It's always the cells which vary. And if you think about it, it's not so surprising. It is a dramatic manipulation of biology from pluripotency to a differentiated cells. It takes several days. It uses numerous factors. All of these factors have some variability from the provider. And then there's a human factor. There's all this. We have a big lab with many people involved in it. So I think we have a good infrastructure, and still we see variability on this side. So I think it's the simple answer is it's Kalkmeister's differentiation. Just wondering, how did you determine if an EHT is successful? Ah, that's easy. Was it just based? No, you're right. It's based on one hand on shape and morphology. So it should remodel, but not too much. But we set a threshold for contractile force, which is, I think, 0.05 millinewton, 50 micronewton. Also, do you think it's possible to reduce the variability of the experimental outcomes by using a stricter standard? Well, the problem is, we thought about it, of course. The problem is, if you compare diseased and control lines and you set a limit for, let's say, force, you might make a systematic mistake by kicking out all the diseased lines. So that's difficult. Ah, here, Jeff. Hi, Jeff. You showed that in addition to the limited predictability of individualized EHTs, they take substantial time. Yeah, is there still potential to leverage your pre-existing EHT data in machine learning classifiers, grouping patients by genotype? I hope so. And we are just about to put this paper together and put everything online. So you are very much invited to use your expertise to work on this data. Are those patients cardiomyopathy caused? Well, maybe I was too quick on that point. Are those patients cardiomyopathies caused by genetic reason? We found in about 40% a clear genetic reason. And that's actually also what you find in literature. Even though so many genes are known, you find in a given patient with dilated or hypertrophic cardiomyopathy only in about 40 to 50% mutation, exactly what we found. What is interesting, though, that we found 2 clearly pathogenic mutations in our healthy donors. And in fact, in one, it turns out that also the MRI showed that this was a beginning DCM. So this patient didn't know, or this person didn't know about his problem, but he had a problem. Which, so what do you envision are the next step to make these EHTs more suitable for patient screening? Yeah. I think the answer might lie in even better standardization, probably automatization, because our group is really, I would say, pretty good and pretty advanced. And these people were great, and there were only 2 hands doing it, or 3 hands. I mean, two times 2. So you can't get much better on that side, but maybe by robotization or something like that. Otherwise, I don't really know.
Do you think part of
Yeah.
it also is related to the cell types there for disease modeling? Like, do you think having combination of cell types that are not just cardiomyocytes and fibroblasts would make a difference in those cases?
They do make a difference. We recently made five cell type EHTs, like cardiomyocytes, smooth muscle cells, fibroblasts, endothelial cells, and macrophages. But man, that's even more variable because of course
Yeah.
you have to do in parallel differentiation of 5 different cell types.
Yeah.
So that's the problem. The more complex we get, the more variability we get into it.
Mhm.
So I'm not sure whether that would really relieve the problem or solve the problem. Do you have any idea of the intra-EHT AP heterogeneity? Intra or inter? I guess inter. As I said, the inter-EHT action potential heterogeneity is very low. It's less than 10%. It's inter-batch, which is a problem. And
Yeah.
whether that is representative of native tissue, I mean, there is native tissue heterogeneity, but I think really, without proving it, that it's mainly the variability coming from the experimental procedures. Which methodology of myocyte differentiation? We used the 2D protocol described originally by Chris Denning. It's a growth factor based 2D protocol, which we call cheese protocol, because it's providing these cheese holes. That is actually very robust. We also use a spinner flask protocol, but that is only good for certain very, very well performing cell lines. To deal with variability, would it help to make multiple clones from each patient so the differentiation is done multiple? Of course, that's the point. Of course, yes. But that's, you know, this project costed 2.5 million euros and 2 postdoc lives and 5 years. And of course, if we would have done 3 clones, 3 batches, systematically, I'm sure we would have got better data. Whether they would have been really different, I don't know. So if you if you want to identify, really evaluate the phenotype of a given cell lines, you have to make several clones, you have to make several batches and large n numbers. Then you get a good answer. But you cannot do this for screening. That's just impossible. It's too expensive, takes too much time. That's actually my point. I really don't take me wrong. I don't say this is an argument against iPSC disease modeling. This data is an argument against the, let's see, somewhat naive idea of some people that you can do a screening with these cells for something like a clinical screening. In my way, no. In my view, that's not possible. Have you compared the electrophysiological properties of EHTs? No, we don't know. But I would say our sharp microelectrode data in EHTs are actually very, very good. And they're better than most things I read about organoids. I never worked with organoids, so I don't know. But it's as good as a native tissue, so it cannot be much better. Does the above average HCM line, yeah, that's an interesting one, show that the diseases is more systemic cell-cell interaction than intrinsically caused by the cardiomyocyte biology of HCM cells itself? I don't know. You have to consider that both in HCM and DCM, these people get normally old. You know, they have a normally completely fine heart. Many of these people are how do you call this? I mean, high-end athletics. You know, these football players dropping dead on the field. These are often HCM. So these people do extremely well, and I think that's what we see here. These people a priori are normal and even maybe a bit super normal, And that's probably in our system a good feature. But that of course doesn't mean that they are super normal on the long term. This HCM is known for hypercontractility, and I think that is somewhat what we see here. That they do even better than normal, but on the long term, that would lead to fibrosis and too much energy consumption, etc. But so in a way, it's quite well compatible with the. We were surprised, but if you think about it, not so surprising.
Exactly. Well, thank you so much for giving a fantastic talk. I know it's getting probably very late over there in
It's getting
Germany.
dark now, yeah.
Yeah. But thank you so much for the wonderful seminar um, and thank you for helping us, finalize the last few uh, tissue talks of the season.
Yeah, it was my pleasure.
Thank you.
Good luck to everybody.